Hello, everyone. Thank you for the introduction and thank you for inviting me here today. It's such a pleasure to be here with the amazing presenters and audience. So my thesis is about the identifying identifying the subtypes in post-concussion syndrome using structural and functional connectomes and relating that to neuropsychological measures. As we all know what concussion is, I just want to highlight that there are a couple symptoms that comes up with concussion, such as anxiety, depression, headaches, and so on. We usually expect these symptoms to resolve within the first 3 months. But 10 to 15% of these patients, unfortunately, show persistent symptoms for a long time, which we categorize as post-concussion syndrome. The criteria that is used in this clinic is DSM-4, which means they need to have an impairment in memory and attention and minimum 3 of the following symptoms that's listed there. But it's really hard to diagnose post-concussion syndrome because the CT and MRI images of these patients are completely normal, and there is currently no biomarker for post-concussion. So what ends up happening is the diagnosis is actually based on self-reports of symptoms. Of course, researchers have been trying to use different techniques to detect subtle changes that you might see in the brain. One of them is white matter integrity, especially in long tracts, such as corpus callosum and anterior coronal, and so on. And the reduced integrity of these white matter tracts have been correlated with executive functioning. At the same time, some people looked at resting state. Resting state is in you get functional MRI in the MRI when you are lying down and not really involving in any sort of task, but just letting your mind wander. Temporal functional changes at the subacute stages and frontal functional changes at later stages have shown in post-concussion syndrome, but it's also related to symptom severity. There are also really few papers who actually looked at the structure function at the same time, but there's this huge gap in the literature of how this structure and the function relates to the cognitive outcomes. And so this is what we are actually researching. So our hypothesis is, can we actually look at the structural and the functional changes in the post-concussion patients, and can we actually subtype patients just based on these changes? What it means is that, is there a group of patients in our post-concussion data that, for example, experience more memory problems versus executive issues, or can we create a scale for post-concussion as in mild versus severe? And I'm going to explain how I do this. Our aims is first to compare the structure and the function in the controls versus the patients, and then try to identify the subtypes in the patients by excluding the controls, and then try to investigate the relationship between these subtypes and the neuropsychological measures. And this is a cross-sectional study. We have 96 subjects with 83 patients, and the age range is from 17 to 67. So I'm not really going to go into the details of this pipeline, but we create connectomes, structural connectomes, based on the Human Connectome Project pipeline. The connectome represents whole brain networks. It's based on diffusion-weighted imaging. The same idea with the functional connectomes, this time it's based on the resting state images. Well, the images look really pretty, but what we analyze are these, the connectivity matrices. Each entry represents the structural integrity between two pairs or functional coupling between the two pairs. Based on these structural connectivities for each person in our data that we created, we also came up with this machine learning sort of pipeline to subtype our patients. Again, I'm not going to go into details. Don't worry, there's not going to be math in there. So the first part is using random forest feature selection. Just because we have a lot of pairs of regions in our data, it's really hard to fit that into any of the models. After, for random forest, feature selection is actually to identify the features that are kind of important in separating the controls from the patients. Once we identify the features, we put those features into Gaussian mixture model, which is an unsupervised machine learning, which means in this case we exclude the controls and only look at the patients and try to find the subtypes. And the model comes up with the subtypes, looks at the data and the distribution of these patients of the structural integrity or the coupling, and at the end creates the groups, and we compare those groups in terms of the number of symptoms, the cognitive outcomes, and so on. Let me go through the resting state data first of all. We did this, again, the same pipeline, the first random forest feature selection. It selects 30 features, 30 pairs of regions, and then we put that, I'm just gonna give you some examples, but not 30 of them are gonna be there, of course. We put those 30 regions into Gaussian mixture model. The best fit of the model is the one with the 23 regions. Not super important, but it separates patients into two subtypes. And then we compare those subtypes. Okay, let's look at what they are. In terms of the resting state analysis, these two subgroups are not significantly different in terms of age, number of concussions, or years of education. So the subtypes are not driven by any of these factors. But what's different between these groups are vision symptoms. Group 1 experiences significantly higher number of vision symptoms compared to the group 2. Well, in this case, you can say group 1 is really severe, group 2 is mild. The other difference was the functional coupling between right lingual and left lingual, which was also significantly higher in the severe group. It might be some sort of a compensation mechanism that we see in those patients. The other one was again, higher activation between left thalamus and left lingual. If we were to compare these to the controls, you will see the severe group is the vision symptoms for severe group is significantly higher than the controls. But there isn't really much difference between the mild and the controls. So the mild group is really looking like controls, actually. The same idea goes with the second. I'm just going to give you the examples for the first two. The same thing goes with this one. The coupling between these groups are actually significantly higher in the severe group compared to the controls, but mild and the controls are showing some sort of similar profiles, which we find really interesting. The same idea goes with the structural connectivity. We're looking at fractional anisotropy in this case. The same idea, random forest was run. We selected 60 pairs of regions, and that was put into Gaussian mixture model, again, to subtype the patients only based on the structural integrity. And then we compare these two groups that came up from the second model. When we look at it, again, age, number of concussions, and mean the years of education are not significantly different between these two groups. But what was different was actually the correct answers on strip color word testing. So group 1 was getting higher scores on this task than group 2. In this case, we are going to say group 1 is mild, group 2 is severe. The second one was the white matter tracks between right cingulate and left pericalcarine. Again, the integrity was better for the mild group. And the third one is just one of the examples between right parietal and right lingual. The white matter tracks was, they had higher integrity in the mild group. If we were to compare this to the controls, you would see severe group is actually not, well, the mean seems to be lower than the controls, but it's not significant. Maybe this task is not super sensitive to the tech, but it's still there. But if you were to look at the second one, you would see the severe group has significantly lower white matter integrity compared to the controls. But mild group is almost the same as the controls. Okay, so I showed you structural connectivity, the functional connectivity, and I said we created different subtypes based on different modalities. What if we were to combine these two modalities? What would happen? In this case, we tried to track down the people who were always in the severe group, both for structure and the function, and there were 16 of these people. And we also tracked down to people who were always in the mild group for both structure and the function. If we were to compare these to the controls, we would actually see different sort of subtypes. In this case, I'm just going to give you one example. What was different in the severe group who had significantly different structural and functional changes showed higher depression scores. These are self-reported sort of symptoms compared to the controls. But again, you would see the controls are just like the mild group. So to conclude that, usually we try to find biomarkers between, or to separate patients from the controls. maybe we are not really successful because there is this mild group who shows similar profiles as the controls, and that is driving our sort of statistical analysis in that way. But if we were to able to find these subtypes and separate them, then we might be able to find better biomarkers. So to conclude, computational models would be able to help us subtype patients. We also show that different modalities and combining them are actually explaining different cognitive domains in post-concussions. And the last thing is, this study is really aim to create precision medicine. And so if we were to able to tell patients what kind of problems they had, we would be able to give them better treatments. And thank you.